Okay, guys, here we are. We're going to start chapter 8 today. This is the title of this chapter is an introduction to metabolism. So before we get started with some of the details of this chapter, we need to talk about some definitions and think about some things that we're going to be talking a lot about throughout this chapter. So the first thing we have to do is define metabolism. So that is your cell's capacity to acquire energy, to build molecules or break apart molecules, and to release substances. So one of the things that we know that your cells are always doing are constantly building and taking apart molecules. So remember all the way back from one of the early chapters, we talked about condensation reactions and hydrolysis reactions. So condensation reactions were where we took little things and put them together to make a big thing, and hydrolysis reactions where we took big molecules and broke them apart to make little molecules. So doing those sorts of reactions requires metabolism, requires energy in your cell. And so your cell's ability to acquire that energy is what we refer to as the metabolism. And then when we talk about energy, you guys have probably talked about these types of energy before, maybe in other classes. Maybe you remember from high school when we define energy, we have to think about the form in which that energy is in. Potential energy, kinetic energy, heat energy, which is sometimes called thermal energy, or chemical energy. And so we'll talk about the differences in these types of energies as we go through this chapter. So one of the things that we know is that there are laws that govern energy and how energy is acquired and how we use energy. And so one of those laws is called the first law of thermodynamics. And this law says that energy cannot be created or destroyed. It can only be converted from one form to another. So we cannot just produce energy out of the environment. Energy doesn't just disappear when chemical reactions occur, but energy is always converted from one of those forms to another form. So from potential energy to kinetic energy, or from chemical energy to heat energy. It's converted. And so a fire is an example of an energy conversion. So we have chemical energy here in our wood. So there is chemical energy stored in the bonds of the molecules that make up the wood. And once we spark that wood with a flame and start a fire, we are now converting that energy to heat, or thermal energy. And that is the heat that is released into the environment. And that's the energy that is released in the environment. So we convert that chemical energy to heat energy through the process of a bonfire. The second law that governs energy is called the second law of thermodynamics. And this law says that energy flows from a highly concentrated area to a less concentrated area. So if you remember, all the way back when we were talking about membranes and diffusion, we talked about the fact that ions in water flow from an area of high concentration to an area of low concentration. Well, energy does the same thing. And so this says that energy in the bonds of molecules, that would be a very concentrated area, and it escapes during conversions to areas that are less concentrated, like the environment. So remember, if we had our bonfire, we had our wood, our log, that had all that chemical energy in there. So that was a high concentrated area. And when it was on fire, all that chemical energy got converted to heat and was released out into the environment. That was an area of low concentrated energy. Another example is the sun. The sun is obviously very highly concentrated with energy. The sun's energy escapes and hits the earth, which is less concentrated with energy than the sun. We never send energy back the other way, right? We never send energy from the earth to the sun. That does not happen. So energy flows from the sun, the area of high concentration, to the earth, an area of low concentration. All right, so we've talked a lot about energy, but we should probably define it before we go much further. Energy is the capacity to cause change. So anytime there is a change, a chemical reaction occurs, something heats up or something cools down, we're talking about energy transfers. So energy, as we just talked about, can exist in several different forms. And some of those forms have the ability to perform work. All right, so let's talk about the different forms of energy. So we have kinetic energy that is associated with motion. So anytime you're moving or something else is moving, there's kinetic energy being released. Heat energy, or that thermal energy, is associated with random movements or atoms of atoms or molecules. So you might have talked about maybe in your high school classes about when you heat a substance up, the molecules move faster and have more of a tendency to bump into one another. And so that is that heat energy or that thermal energy. Anytime we apply heat to something, the molecules speed up, they move around faster. Potential energy is an energy that is based on location or structure. So we oftentimes think a lot about potential energy as being determined by a location of an object, and I'll show you a picture of that in just a second. We've already talked a little bit about chemical energy, so it's kind of a form of potential energy, but it's available in the chemical bonds of a chemical substance, and it can be released in a chemical reaction. So almost every chemical substance has some form of, or some amount of chemical energy stored in the bonds of the molecule. And when we break that molecule apart, we can release that energy into the environment. So remember, the key is that energy cannot be converted or cannot be created or destroyed, but it can be converted from one form to another, from any of these one forms to any of the other forms. Okay, so here's a picture kind of talking about the difference between kinetic energy and potential energy. So if we look at the diver up here on top of the platform, he has very high potential energy up here because of his location. So if he gets to the edge of this platform right here, he is going to have a great deal of potential energy because it would be very easy for him to dive and off the end of the platform. So when he's up on this platform, especially at the edge, he has a tremendous amount of potential energy. Once he jumps and is flying through the air, that potential energy has been converted to kinetic energy because now he's moving, and so that is that energy of motion. Once he hits the water, he has less potential energy. He's still probably moving, but he has certainly less potential energy than he did at the top of the platform. As he climbs up the steps to do it again, he is converting the kinetic energy of his muscle movement to potential energy. So as he's climbing up, he's using that kinetic energy to climb, but it's becoming more and more potential as he gets to the top of the platform, because at the top of the platform, he's got all that potential energy stored up based on his location. Okay, when we talk about energy, we generally talk about it in the terms of this letter G, and G stands for Gibbs free energy. So there was a man named Gibbs who studied energy a lot, and so he came up with this equation. And so we talk about free energy, which is the amount of energy available to do work in a system. So it's sort of like your available balance in your checking account. So for example, if you have $100 in your checking account and you go shopping today and you buy a $50 pair of jeans, when you look up your balance as soon as you leave that store, it's going to say you have $100, but you only have $50 available because the $50 has already been sort of pulled off that you paid for the jeans. So Gibbs free energy is the amount of energy available to do work. This is sort of like that available balance in your checking account. That other $50 is still there, but it's no longer available for you to spend. And so that's how it works with energy too. Some of the energy within a system is being used for other things and is not available to carry out the work of the system. So the way that we calculate G is we take enthalpy, we take total energy, which is H. So that's our total amount of energy. That would be like the $100 in our checking account. we take our total amount of energy, we minus Ts, where T is temperature, always in Kelvin, and S is something we call entropy. So entropy is a little bit of a hard concept to get your head around, but entropy is amount of disorder. So basically the universe is always sort of moving towards a state of disorder, so more and more randomness. And so that is what we mean by entropy. It's a measure of the randomness or the disorder in the universe. And so when we carry out this equation, H minus Ts, we get G, which would be like the $50 in our checking account that's left over after we bought the $50 pair of jeans. So again, that is like our available balance of energy to do work. Okay. So in reality, when we talk about G and Gibbs free energy in the lab, in theory, we talk about changes in G. So that's what that delta refers to, is change. We very seldomly calculate just plain old G, but in theory, in the lab, we calculate delta G. And so the way that we calculate delta G is we take the G of our products, or our final position, minus the G of our reactants, or our starting position. And so if that delta G is a negative number, we say that that reaction is spontaneous, that it would happen all by itself, that we wouldn't have to supply it with energy or anything, and we call that an exergonic reaction. If, when we do this calculation, G final minus G starting, or G initial, and we get a positive number for delta G, that reaction requires energy. We would have to supply some energy, usually in the form of heat, to make that reaction go, and we call that an endergonic reaction. So we'll talk more about those in just a minute. Okay, so if we look at some diagrams here, this is what we call free energy diagrams, and this is going to represent exergonic reactions versus endergonic reactions. So we're going to do some calculations, and we're going to assume that this line right up here at the top of our graph represents 100. And so this line down here, where our products are, we're going to say that that represents about 25. And so remember, for our delta G, we're going to take the G of our products minus the G of our reactants. So if we look at this first energy diagram in orange here, if we were to do this calculation, we would get the G of our products is 25 minus the G of our reactants, we said was about 100. And so we get a negative number there. We get a negative 75. And so what that means is that there is energy released when our products are converted to reactants. We get about 75, what we call kilojoules of energy released. And so this is what we would call an exergonic reaction. So that delta G is negative. That means that this reaction would happen whether or not we supplied energy to it, and those reactants would spontaneously convert to products, releasing energy as they did so. If we look down here at the blue graph, so remember this line up here, we're going to say it's about 100, and this line down here we're going to say it's about 25. So if we look at this reaction, we start with our products, they're at about 100, minus our reactants at about 25, we get a positive 75 joules. And so what this means is that we have to supply energy to this reaction. We have to take those reactants and we have to supply some energy, again, usually in the form of heat, in order to make those reactants go or be converted to products. And so energy is not given off in this reaction, but energy is applied to the system in order to make it go. And so we refer to that as those endergonic reaction. So delta G is positive, and that is what we call an endergonic reaction. Okay, so we need to refresh our memory a little bit about chemical reactions before we move forward. And so this is just a general reaction, X plus Y yields XY. So remember, X plus Y, we refer to those as our reactants. XY is going to be our products. And when you see these double arrows like that in the center, that tells you that that reaction is reversible. So we can have X plus Y going to XY, or we can break down XY into its individual components, X plus Y. So one is the forward rate, and one is the backwards rate, or the reverse rate. When we talk about reactions going to equilibrium, that means that the forward reaction is equal to the reverse reaction rate. It has nothing to do with amounts of reactants or products. It has to do with reaction rates. So at equilibrium, the forward rate is equal to the backwards rate. So don't get confused by that. A lot of people get messed up and think that equilibrium means that the amount of reactants or products is the same, but it has nothing to do with amount. It has to do with rates. And we're thinking about the forward rate being equal to the backwards rate, and that's what happens at equilibrium. So if we were to increase the amounts of reactants, if we had a whole bunch more X plus Y over here, we would increase the forward rate. If we had an increase in the amount of products, so if we had more XY over here, we would increase the reverse rate. And so that's the way we can sort of shift the equilibriums and shift the rates, is by increasing the amounts of products versus reactants. Now remember, we have laws that govern chemical reactions, just like we have laws that govern energy transfers. And so the law that governs the chemical reaction is the law of conservation of mass. So this law says that matter cannot be created or destroyed. So what that means is when we are looking at chemical reactions, we have to have the same number of atoms of each element on both sides of the reaction. So if we're looking at our sort of generic reaction here, over here on the reactant side, we have 1X. So we better have 1X over on the product side, and you can see that we do. And over here on the reactant side, we have 1Y, so we better have only 1Y over on the product side, and you can see that we do. So this is what we call a balanced chemical equation. So we can have much more complicated ones. If we look at this one, this is the conversion of carbon dioxide and water to glucose and oxygen. So you might recognize this as the reaction of photosynthesis. This is the reaction that plants carry out every day when they make glucose. And so if you look at this reaction, I'm just going to go through a couple of these, but if we look over here on the reactant side, you can see we have 6 atoms of carbon. And so if you look on the product side, you can see we also have 6 atoms of carbon. Over here on the reactant side, we have 6 times 2, which is 12 atoms of hydrogen. And over here on the reactant side, we also have 12 atoms of hydrogen. So this is again what we call a balanced chemical equation, and this is abiding by the law of conservation of mass, saying that we have the same number of atoms of each element on both sides of the reaction. Okay, so when we talk about metabolic pathways, there are a couple of players involved that we need to talk about and we need to define. So the first one are enzymes. We know that most reactions that happen in your body happen far too slowly to sustain life without the use of enzymes. So if we didn't have enzymes in our cells that were speeding up the chemical reactions that were happening in our body, we would not be able to sustain our life. Every single reaction in our cell would take far too long and require far too much energy for us to be able to live without the use of enzymes. So enzymes speed up chemical reactions, they make reactions go faster, and usually every single metabolic pathway in your cell, every single reaction within a metabolic pathway is controlled or regulated by enzymes. So we need to talk about enzymes and think about how they work. The other player in metabolic pathways are cofactors. So cofactors help enzymes. They're sometimes referred to as enzyme helpers. So their job is to carry electrons or hydrogen ions around the cell and deliver them where they need to be. So we're going to talk a lot more about cofactors when we talk about photosynthesis and cellular respiration in the upcoming next 2 chapters. The other player in metabolic pathways is one that you're already familiar with, that we talked about way back in chapter 7 when we did the membrane chapter, and these are the transport proteins. So their job is to carry substances across the cell membrane, either towards or away from metabolic reactions, depending on what transporter we're talking about. So remember, these were proteins that were embedded in the plasma membrane, and they allow certain molecules to pass through the membrane. All right, so let's talk about sort of the other big player within a metabolic pathway, and that is ATP. So this is your cell's energy currency. This is the way that your cells use, spend, and store energy. Okay, so ATP consists of one adenine base, so that is a nitrogenous base that we talked about when we did our chapters on DNA and RNA. So that is your adenine there, highlighted in blue. It has one ribose sugar, so this is highlighted in pink here. And again, that should look familiar to you from our chapter on RNA. So that's the same sugar that's present in RNA, so that's your ribose sugar. And then we have 3 phosphate groups. So ATP stands for adenosine triphosphate. So that's where the 3 phosphates come from. So adenosine triphosphate. So we have the adenine, the ribose, and then 1, 2, 3 phosphates. Again, ATP is the way that your cells store, use, spend energy. So when we talk about your cell's ability to acquire energy, we are talking about your cell's ability to make ATP. And we're going to talk a lot more about ATP when we go through cellular respiration in chapter 10. But this is the way that your cells use their energy. Just like you use money, that's the way that you spend money and they buy the things that you want. Your cell's money, so to speak, is ATP. So the way that we talked about, remember that almost all chemical molecules have energy stored in the bonds that make up that molecule. Well, ATP has a lot of energy stored in its bonds, particularly in this bond right here between the 2nd and 3rd phosphate groups. When we break that bond, when we break off this 3rd phosphate, we release a tremendous amount of energy. And so that energy can then be applied to metabolic pathways or used within the cell to carry out other chemical reactions. So this is a good example of the chemical energy being stored in a bond and being released within the environment. All right, so let's talk a little bit more about enzymes. So again, these are one of the major players of metabolic pathways, and there are a couple of features of all enzymes that we need to be aware of. So nearly all enzymes are proteins. Remember, we talked about the molecules of life way back in chapter 5, I believe. We talked about lipids, carbohydrates, nucleic acids, and proteins. Well, enzymes are a special form of proteins. So that is where they fit into sort of the molecule of life structure. Remember, their job is to speed up chemical reactions. They are not consumed during a chemical reaction, but they can be reused over and over and over again to catalyze the same chemical reaction multiple times. And typically we use the same enzyme for the forward and reverse reactions. So remember, we talked about a reaction that's able to go forwards and backwards. And we can use the same enzyme to catalyze the forward reaction that we use to catalyze the reverse reaction. The other thing we know about enzymes is the way that they work is they decrease something called activation energy or collision energy, which allows a reaction to occur. And so I'm going to show you a picture of this in a second. But the way that they do this is they bring the reactants together and they orient those reactions in such a way that they can react and be converted to products. And this occurs at a site on the enzyme that we call the active site. So the reactants come together at the active site, they're oriented properly or arranged properly in a specific situation where they can react and be converted to product. And so here we have a graph showing, again, this is an energy diagram showing you our reactants to our products and how we get from one point to the other. So this is showing you activation energy. And so without the enzyme, this is showing you the level of activation energy first without the enzyme. That's the red line. And this is the minimum amount of energy needed for a reaction to proceed. So you can see we have this big hump that we have to get over. So all of this from here, starting at our reactants, all the way up to the top of that graph, that is our activation energy. So that is how much energy we would have to apply to these reactants to get them to convert to products. Once they get to the top of the hill, it's sort of like they're on a roller coaster, and it wouldn't take much to just sort of push them down the hill, and then they can be converted to products. But this area here, from where the reactants are to the top of this hill, is like going up the hill of the roller coaster, and we have to supply a lot of energy to push that roller coaster car up to the top of that hill. Now, with enzymes, what they do is they lower that activation energy. So you can see now our activation energy is much shorter. We only have to get here to the top of this little blue line, and then we can go down the hill like we're on the roller coaster to be converted to products. So enzymes lower that activation energy, lower the amount of energy that we have to supply to that reaction to get to the top of the hill. So one way that I like to describe enzymes, if you ever tried to quit smoking, so you might have used like a nicotine patch or nicotine gum. Those are things that sort of get you over the hump to the top of the hill. They help you get there. So you have this huge hurdle that you have to get over in order to quit smoking. And so it's sometimes hard, right, to get to the top of that hill. It's hard to get over those hurdles. But if you have things like nicotine gum or nicotine patches, they lower that hurdle and make it easier for you to get over here to quit smoking. All right, now enzymes don't always work. They have optimum conditions at which they work best. And one of the factors that affects enzyme activity is temperature. So temperature or enzymes can easily be denatured or destroyed if temperatures get too low or too high. So most of the enzymes in your body work best at 37 degrees Celsius because that is your body temperature. That is the optimum range for your enzymes to function at their maximum capacity. If temperature gets much below 37 or much above 37, those enzymes are going to lose that optimum activity and they're going to be destroyed. So we have to keep those enzymes around 37 degrees Celsius so that they function the way they're supposed to. The other thing that can affect enzyme activity is pH. And so different cells in your body have different pHs, and the enzymes within those cells function best at those optimum pHs. So for example, the enzymes that are in the lining of your stomach are going to function best at a pH around 2.4 because that is the pH of your stomach. It's a very acidic environment. And so if the acidity or the pH of your stomach shifts much below 2.4 or much above 2.4, those enzymes are going to be denatured and destroyed and not working as they should. The enzymes in your bloodstream, for example, are going to function best at a pH around 7.4 because that's the pH of your bloodstream. If the pH of your blood gets much below 7.4 or much above 7.4, those enzymes start to lose activity and would eventually be destroyed or denatured and not work as they should. Now, I want to pull this all together sort of here in the last few slides and think about how do we relate these endergonic and exergonic reactions to the condensation and hydrolysis reactions that we talked about much earlier in the semester. So first of all, let's think about those endergonic reactions. These are reactions that have that positive delta G. So remember, delta G is G of the products minus G of the reactants. So these are reactions where the products have more free energy than the reactants. So these would require energy. We would have to supply energy to these reactions to convert reactants to products. And these are going to be your condensation reactions that we talked about earlier in the semester. So remember, these are the reactions that take small things and put them together to make big things, all right? So we take small molecules, put them together to make large molecules. The exergonic reactions, remember, these are the ones that are going to have negative delta G's. So the reactants are going to have more energy than the products. These are going to be your energy releasing molecules or your hydrolysis reactions. So this is when we take those big molecules and we break them apart to make little molecules. So this is breaking things apart. These are your exergonic reactions. So we need to be able to make connections between those types of reactions and whether they are endergonic and exergonic with their positive delta G's or negative delta G's. Now, I want to go back just a second to this idea of metabolism and remember the fact that all of these reactions within a metabolic pathway are controlled by enzymes. So again, this is your body's way or your cell's way of acquiring and using energy. So if we look at examples of some metabolic pathways, we need to compare that to just one simple reaction. So typically most of the reactions that we have talked about sort of look like this. We have reactant A, which is going to go to product B. It may be controlled by enzyme E, and it's going to go back and forth until it reaches equilibrium and the rates are the same. Metabolic pathways are a little bit different. So these are sequence of enzyme-controlled reactions where the product of one reaction becomes the reactant of the next reaction. So here we might have reaction 1 where we have reactant A going to product B catalyzed by enzyme 1 or E1. In our second reaction, that B that was our product now becomes our reactant, and that is going to produce product C. And then in the third reaction, that product C now becomes reactant C, and it's going to be converted to product D. And then in the fourth reaction, that product D is going to become reactant D and be converted to product E. And so that's what we then call our end product, and that can either be used or stored or whatever it is that we need to do with it in the cell. So this is where you have a series of related reactions where the product of one step becomes the reactant in the next step. And this is what we refer to as a metabolic pathway. Now, metabolic pathways, we can have two different types. We can have what are called biosynthetic pathways or degradative pathways. The biosynthetic ones do exactly what they sound like they would do. They make things. All right, so anytime you synthesize something or carry out a synthetic reaction, you are making something. So these are reactions or pathways that result in making a final product. So these are likely going to be condensation reactions. They're going to be endergonic. You're going to have to supply energy to these. The degradative pathways, their job is to break things. So these are breaking things down that we no longer need within the cell, taking them back down to their original components. These are your hydrolysis reactions. These are typically your exergonic pathways. These are going to be reactions that release energy as we break those things apart. All right, so let's go back to the idea of enzymes for just a second and think about, remember we talked about the fact that enzymes are not always functioning at their optimal capacity. So we talked about things like temperature and pH that can affect enzymes, where you can also have inhibitors that affect enzymes activity. And so we can have three different types of inhibitors. We typically talk about reversible inhibition. And so this is where a inhibitor or some sort of molecule is going to bind to that enzyme and prevent it from doing its job, prevent it or slow it down from speeding up a chemical reaction. So inhibitor forms a weak bond with the enzyme, and a reversible inhibition can be competitive or non-competitive. And what we mean by reversible inhibition is that inhibitor can bind or it can detach. So just because it binds doesn't mean it stays bound. It can reverse and detach from that enzyme. So with reversible inhibition, you can have inhibitors that come and go. Now, when we talk about, there are two types of reversible inhibition. We can talk about competitive inhibition or non-competitive inhibition. I'm going to show you a picture of this in 1 second. So a competitive inhibitor is an inhibitor that competes for or competes with the normal substrate for the binding of the active site of the enzyme. So this is a molecule that sort of looks a lot like a substrate and sort of tricks that enzyme into thinking that it is the substrate. And so the enzyme will bind it, and it turns out to be a competitive inhibitor. With a non-competitive inhibitor, that is a molecule that binds at a site other than the active site. So it's not competing with the substrate, but it still binds to the enzyme and prevents it from doing its job. So if we look at a picture of this, I think it'll be helpful. Okay, so here we have our enzyme in blue. We have our substrate here in green, so that's substrate number 1. And we have substrate number 2 here in orange. So this is a 2-substrate reaction where we have to have both of those substrates bound to the enzyme in order for that enzyme to do its job. So if 1 and 2 both come in and bind to that enzyme is going to speed up that reaction. It's going to bring those substrates together so that they can orient themselves properly and be converted to product. However, in this case, we have inhibitor here in purple, and that inhibitor is going to compete with that active site right there that I've circled in green. So that's where that green substrate number 1 would typically bind. And so the purple inhibitor is competing with that substrate for that active site. So if the inhibitor binds, the substrate cannot. The site is blocked. And so that inhibitor has bound to that active site, and now it's not going to let the enzyme do its job. Now remember, we said this is reversible inhibition, so that inhibitor could leave, and once it detaches, the substrates could bind and the enzyme could function. So it's not permanent inhibition, it's temporary inhibition. So that's competitive inhibition. When we talk about non-competitive inhibition, so we still have our enzyme here in blue, we still have our 2 substrates in green and orange, and they still have their active sites or their binding sites for that enzyme where they need to interact. Now here we have an inhibitor out here, again in purple, that is going to bind to that enzyme, again at a different site. It's not binding to the active site like the first one was. This is binding to that enzyme back here in the back. But when it does that, when the inhibitor binds, look, it changes the shape of the enzyme. So now our substrates have lost their active site. Those binding sites are not there anymore. And so now the substrates can still not bind to that enzyme. The enzyme still cannot do its job to speed up that reaction. Again, this is only temporary, so this inhibitor could leave, and when it does so, the enzyme is going to go back to its original shape and the substrates can bind. So the idea is, or the key is, competitive inhibitors bind to the active site of the enzymes. They compete with the substrates for binding to the active site. Non-competitive inhibitors bind to a site other than the active site on the enzyme. They bind somewhere else on that enzyme, but they change the shape of the enzyme such that the original substrates can no longer bind. All right, so that is the end of Chapter 8. I will see you in class.